We have spent a lot of time discussing proteins, structure, and function. They come in primary structures, secondary structures, tertiary structures. I'm going to be carrying it with me. Like my car, like the homeless car. I'm going to buy the spinning one. Don't worry, I'm going to buy the spinning one. Okay, all righty. So let's begin. So up to this point, what do we know about proteins? Proteins are macromolecules. It's not a secret anymore. It's not micro. It's not micromolecule. It's a macromolecule. And we have different types of macromolecules. Proteins, nucleic acids. Then the next unit, you're going to have carbohydrates and lipids. Now, with proteins, we know that all the macromolecules are built up from building blocks. What are the building blocks for the proteins? What do we call it? Amino acids. And we said we have different types of amino acids based on the R substituent. If the R substituent is acidic, the whole amino acid is acidic. If the R substituent is basic, the whole amino acid is basic. If the R substituent is a cation, the whole entire amino acid is going to be positively charged. If it's an anion, the entire amino acid is going to be negatively charged. So the R group is what defines the properties of all the amino acids. Now, we also learned that even the protein folding, how is the protein going to fold to become a globular structure? How is the protein going to become a quaternary, tertiary, secondary, primary structure? It's all based on the primary sequence and the R groups. And then we learned about the different interactions that give us those different globular proteins. Sometimes we have covalent bonding. Other times we have salt bridges, disulfide bridges. Sometimes we have hydrophobic interactions. So different interactions are like a code that is what make the proteins unique. So those reactions are based on the enzymes. The enzymes, without them, the reaction is going to happen, obviously, eventually, but the reaction is going to happen in thousands of years. The reaction is going to happen in thousands of years. So what is the enzyme going to be doing? The enzyme is a catalyst. It speeds up the rate of the reaction. And today we're going to look more into the kinetics of how exactly does the enzyme speed up the reaction. So now if you want to, there are different factors, different very important factors that affect the rate of the reaction, how fast the rate of the reaction going to go. Let's say you have a reaction of multiple reactants reacting with each other to give you one product, or multiple reactants come together to give you multiple products. Well, how can that pace? How can we increase the pace? So if we have, it doesn't fall. If we have a, be patient, we're gonna make it. Okay, so if we have a single reactant, that's a single reactant that's going to give you multiple products, right? B plus C. That's an example of a decomposition. A decomposed into B and C. That's a single reactant that is going to give us multiple products. Sometimes you can have a single reactant to give you a single product. Sometimes one single reactant gives you multiple products. That's one type of a reaction. And sometimes you have a single reactant giving you a single product. Sometimes you have multiple reactants, A plus B, those are the reactants, giving you a single product. Those are multiple reactants giving you a single product. Or you can have multiple reactants giving you multiple products. But in the end of the day, no matter which reaction we're dealing with, we need to go from reactants to products. And if you remember in our thermodynamics, we have learned that our y-axis is representing the free energy, delta G. That's the energy we look at when there's an exergonic reaction. Is it minus? If it's minus, so you know the energy is coming out exergonic. If it's plus, that means the energy is endergonic. The energy has been stored into the bonds. And on the x-axis is going to be the timeline. Well, we know that if you're going from reactants and you go into product, you have to climb the hill. You see the products are always more stable. They have less energy. If you look at this y-axis, the more you go up, the more delta G increases. So reactants always have a lot of energy. The reactants are always bubbling with energy and unstable. They're like little kids with ADHD. They're running all over the place. But products, they are so mature and they're lazy and they are slow. These are the products. So products are stable. Reactants are unstable. And products are stable. Well, in the end of the day, if you're going from reactants to product, there is an energy barrier here, an energy hill that you need to surmount. You need to surmount this energy barrier in order to go from reactants into products. Are we clear? What's the name of this energy hill that we need to surmount and we need to cross? What's the name of that? Yes. Excellent. That's the activation energy, and we refer to that as EA. EA is the activation energy. Now, if you look from the reactant line all the way down to the level of the products, what do we call that energy? Is that activation energy? The energy from the reactant line, from the reactant here down to the product. There's two energies here. There's an energy from the reactant side up until the transition state complex, where the asterisk is. This is known as the transition state complex. This transition state complex is the intermediate complex. Let's say we have A plus B is going to, let's say I have AB is going to react with C, and in the end, I'm going to have A separate and B and C together. What do we call this type of reaction? Exchange. Excellent. That's an exchange reaction because A was bonded to B, and in the end, A separated from B, and now B is bonded with C. That's like an exchange reaction. So what happens in the transition state complex? In the transition state complex, you have a half and half. So you have A in like a weak bond with B. So the original strong bond between A and B is beginning to break up. And a bond between B and her new friend C is starting to build. So you have a 50%, 50-50. When 50% of the bonds, because originally it was like that, A and B are bonded, and in the end, you're supposed to have B and C bonded. In the transition state complex, you have 50% of the bonds between A and B originally are broken, they're broken. And you have 50% of the bond to be made between B and C are now beginning to form. So 50% are lost, minus, and 50% are beginning to be formed. So you have a complex that is a half and half. It has a weak bond to the old one, A, and it has a beginning of a bond with C. So it looks like it has three relationships at the same time. Are we clear? The transition state complex is half cooked. It's in the middle between where you used to be, how you used to look like, and how you end up in the end. So the activation energy is the energy from the reactant up until the transition state complex. You really need to surround that energy in order to become product. Are we clear? Questions? Anybody has a question? Okay. The transition state complex is a complex in a special type of state. It is not a reactant, but it's not a product yet. It is half cooked. Because in the beginning, we're supposed to have, don't be preoccupied with writing, look at me, understand, then write in your own words. The transition state complex, for number one, it's a complex. It's a substantial chemical complex. It exists in a special type of a state called transition. It's transitioning. It's transitioning from the state of being a reactant, so it doesn't make sense that it looks like a reactant, because it's transitioning. But at the same time, it's also transitioning to become a product. So if you take out from the beaker a transition state complex, is it going to resemble the reactants, or is it going to resemble the product? Or is it going to resemble neither? It's going to resemble neither, because it's in the middle, where you will see 50% of the bond that used to be between A and B are now broken. Because what is our reaction trying to accomplish? Our reaction is trying to accomplish an exchange where we break A and B and we form B and C. So half of A and B is broken, minus, and half of B and C is being made, plus. That's your transition state complex. So the energy from the reactant up until the transition state complex, we call that activation energy, EA. Some students, if you don't write it, they will just keep it like that way. EA means activation energy, or the energy of activation. What about the energy that is between the reactant and the product? What do we call that? This is the free energy. G stands for Gibbs. That's the guy who discovered it. So the Gibbs free energy is the energy, the smallest route from your reactant to your product. Remember in the past I told you if you want to go to Staten Island now, you got to take the subway and then take the bus. So that's like going to a transition state. That's like going through a really big hill. But if you call an Uber, the Uber is going to be like an enzyme. It's going to take you there by reducing the long route. So the enzymes, they lower, what do enzymes lower? They lower the activation energy barrier. So that it's no longer a hill, it's like a little bump. So when they lower the activation energy barrier, do you get your products faster or slower? you get a lot, much faster. But in the end, whether you're looking at the MTA, the subway, and the bus, or whether you're looking at an Uber driver, in the end, they can only reduce the long route, but can they reduce the minimal distance between your location A and B? Absolutely not. Same thing. The energy between the reactants and product, this box here, reactants and product, that can never change. That's called the free energy. The free energy of GIBs cannot change. Only the activation energy change. So if you want to make a reactant like these guys go faster, you want to make a reactant like these guys go faster, one of the factors is to add an enzyme. Once you add the enzyme, what happens to the activation energy barrier? Drops significantly, reaction goes faster. You're going to have a lot of products in no time. So that's one factor. What about another factor? How about the temperature? How about when you heat up the reaction? Does it go faster? Absolutely. Because what is temperature? Temperature is the average kinetic energy of the molecules. Temperature is the average kinetic energy, meaning the movement, the velocity of the molecules. So if you heat things up, they're going to go faster. So reactants are going to become products much faster. Imagine you combine both. You add an enzyme and you heat up the reaction. Now the reaction is going to go to completion. There's another tricky one, and that's the catalyst. The catalyst, I'm sorry, not the catalyst, the concentration. So they realized that if you look into this graph of this is concentration. Concentration of who? Reactant or product? Reactant. We're trying to get product. So you cannot affect the product. You want to affect the reactant to make it into a product. And so on the x-axis, you have the concentration of the reactant. And on the y-axis, you have the reaction rate. Reaction rate. Or the speed. The speed of the reaction. So they realized something interesting. You have to be able to read this graph. On the x-axis, you have the concentration. So the more you go in this direction, the more reactants you're passing. So here you have added 5 millimoles of reactants. All these guys, you had 5 millimoles. Here, 10 millimoles. Here, 15 millimoles. Here, 20 millimoles. Here, 25 millimoles. Here, 30 millimoles. Here, 35 millimoles. 40 millimoles. 45 millimoles. Going on. If you're going up, you're interrupted. If you go to reaction, how fast is it? How much product per minute? So now we're looking at the speed. How many products are made per minute? So here I have 10 millimoles of product are formed per minute. 20 millimoles of product per minute. 30 millimoles of product per minute. 40 millimoles of product per minute. And you're going up. So you know that in the Y, you have the rate of how much is being formed per minute. You know they have the Nathan hot dogs, 10 hot dogs per minute, 20, that's the rate. Whenever you have something per unit time, what is speed? Speed is a rate. You go 20 miles per hour. That's a rate. When they say rate is always something per unit time. Okay, so the rate is something per unit time. You know they used to pay professors an hourly rate for lecture. So you teach 5 hours, you get paid for each hour. So a rate always has time based on what you do. You're eating hot dogs per unit time. You're teaching lecture per unit time. You're forming products per unit time. So what did they realize? They realized something interesting. That the more you add concentration of reactant, the more the reaction rate speeding up. The more you add, the more the reaction rate speeding up. The more you add, the more the reaction rate speeding up until you get to a certain point where it no longer matters how much you add. It's now also with the temperature. If you heat up the reaction crazy, they're gonna denature. Obviously, you need to moderately heat up the reaction because if you heat it up too hard, you're gonna denature all the enzymes. And also the enzyme. The more enzymes you add, the more reactants you're gonna get. Also with the concentration. So think of it this way. You know sometimes they have shuttle buses. The shuttle bus is making products, taking you from the train station to your home. So now there's like 10 shuttle buses outside the subway station. 10 shuttle buses. But they cannot move because nobody came out the subway yet. So now 10 people come out, 20. The more people come out, the more shuttle buses move. Now let's say 10,000 people come out. No, you're gonna reach a point where the more people come, it's not gonna affect no more because all of the buses are full and used. Same thing. In a reaction, the more products you add, the more reactants you add, the more product you're gonna get. Unless you add so much product that we don't have enzymes to bind to them and convert them to product. So now you're gonna reach this steady line known as the sealing effect. Sealing effect. You're gonna reach the ceiling. So now at this point, before the more the better. The more reactants you add, the faster the reaction rate. The more reactants you add, the faster the reaction rate. The more reactants you add, the faster reaction rate. Until you get a point where it's a steady line, it no longer matters how much reactant you add. Yeah.
Is there a common threshold?
So it depends on which reaction we're looking at. So this is a general idea. It depends on the enzyme we're looking at. So, for example, hemoglobin is an protein enzyme. You saturate, you take a deep breath, the more oxygen in the lung, the more, they're like shuttle buses, the more the hemoglobin is going to pick up oxygen. If you have too much oxygen now, your body is overtly, you have hyperoxemia now, that is not going to affect the rate of the hemoglobin saturation. Okay, so it depends on which enzyme. So here we're now looking at a particular enzyme of choice. We're looking at the whole concept that in every chemical reactions, the more you heat up, the faster the reaction will go. How much the temperature? For some enzyme, you have to heat it up 70, some 80, some 60, some 20. But the baseline is the more temperature, the faster. The more enzymes, the faster. How many enzymes we add? How much milligrams of enzymes should we add? It depends. Also, reactants. How much reactants should we add? How much reactant will we stop? We don't know, but all of them are based on this. Okay. Everybody understand that? Concentration, temperature, catalyst. So it's not the more the better. It's the more the better to some point. Then after that, is not no longer the more the better. Okay. So all of these reactions there, single reactant giving you multiple products, multiple reactants give you single products, all of these different types of reactions there, they're all serving your metabolism. Remember we said metabolism is a sign of life. Metabolism is a sign of life. If you are able to perform metabolism, you are alive. And metabolism is divided into anabolism and catabolism. Catabolism, breakdown of energy so the energy can come out, exergonic. So I can use it to run the marathon. Yes. Yeah, I'm coming back to that. Thank you so much. Anabolism is energy coming in, endergonic, trying to save the energy by storing it into the bonds. Now, the process of storing the energy is also reliant on Leo says Ger. Remember Leo says Ger? Lose electron oxidation, gain electron reduction. Leo says Ger. If you lose the electron, you're oxidized. Which agent are you? If you lose electrons and you're oxidized, which agent that makes you the reducing agent, because you cannot be two in one. You cannot be oxidized and the oxidizing agent. You gotta be, if you're oxidized, you have to be the reducing agent. If you're reduced, you have to be the oxidizing agent. Reduction. Any chemical reaction in which a molecule gains electrons and it will become the oxidizing agent. You know, we discussed the functional groups. That's when we were doing the R substituents. So it's very important to know that the amino groups, there's an amino group and a carboxylic group. That's how we make a peptide bond. When the amino group donates an H and the carboxylic group donates an OH, and now you have water coming out. Phosphate. Phosphate is the basic bond for nucleic acids, as we will learn today. The bond between proteins is known as a peptide bond, and the bond between the proteins is known as a peptide bond, and the bond that connect nucleotides to each other covalently, what do we call that? We call that the phosphodiester bond. The phosphodiester bond. This is some work. I should have listened and bought that moving thing. You know, I never listened. I will listen now. I will go buy immediately. I hope it works. Phosphate. Okay. Protein is a polymer. Did that say monomer? No. Monomers come together to give you a polymer. And as we said, there's an amino group and an R carboxylic group. And the R substituent is what really gives the protein its identity. So as we see here, we went over that. Methionine, that is always the very first amino acid in the translation. In eukaryotes, what about bacteria? Do bacteria not need proteins? Do bacteria not need proteins? Everybody needs proteins. And the proof for that is bacteria do not have organelles. Bacteria do not have endoplasmic reticulum. Bacteria doesn't have a nucleus. Bacteria does not have endoplasmic reticulum. But do they not have a ribosome? They have a ribosome because ribosomes are essential for life. So the only difference is between us and the bacteria. Our first start codon is going to code for methionine, while bacteria codon is going to code for formulated F-met. So we have met, M-E-T, methionine. They have formulated methionine, F-met. F-met. Is that clear? We really need to know this distinction. For human beings, for eukaryotes in general, not all the human beings, is methionine. For bacteria, it's formulated methionine. And you look at all these different amino acids, the only difference is the purple part, which is the R substituent. Peptide bonds, we discussed that. You see, that's your peptide bond. You need to know who donates what. The hydroxyl is coming out from amino acid 1, and amino acid 2 is donating the amino group is donating the H. So that's your peptide bond formed. We talked about the denaturation, and that is in extreme conditions. What causes the protein to roll back and denature? When we say denature, which bonds are broken? The covalent bonds are broken? No, the covalent bonds are not broken. Who is broken? What kinds of bonds are broken when the protein denatures? Hydrogen bonds, excellent. Those dashed hydrogen bonds are broken. But the strong line bonds, the covalent bonds, are never broken. Talked about primary, secondary structure, tertiary, and these disulfide bridges always need sulfur. Sulfur in order for us to make a special type of bond known as a disulfide bridge, and that plays an important role in the folding, proper folding. Quaternary structure is when you have a polypeptide of multiple globular units that are coming together, bonded with hydrophobic interactions. hydrogen bonding, disulfide bridges, ionic salt bridges. That's the amino acid. That's the alpha helix. And there's a tertiary structure. Who remembers, who can give me an example of a tertiary structure protein and a quaternary structure protein? Tertiary and quaternary. Excellent. Myoglobin is tertiary or quaternary? Tertiary. Tertiary. Quaternary. Excellent. Excellent. Okay, proteins cannot stand alone. They need inorganic. What is inorganic? Inorganic means that it's a chemical compound that lacks what? Carbon and hydrogen. So something like phosphate, like iron, that's inorganic. Inorganic. So that's called a prosthetic group. The prosthetic group is like a side battery. It's like a battery. It gives battery to the enzyme to enable it to work. So hemoglobin has 4 prosthetic groups. What are they? The 4 prosthetic groups in hemoglobin. Heme, the iron. Remember I told you, the prosthetic group is always an inorganic non-amino acid moiety. It's like an attachment, an auxiliary, a supplementary part that is attached to the amino acid. And we talked about the importance of the enzymes. Obviously, all enzymes are proteins, but they have like a little prosthetic attachment. You know, human beings, sometimes they wear a wig. Human beings wear a prosthetic leg. People do that. They add things. Enzymes too. They wear a wig, but that is actually like a battery. It's more important than the wig. Actually, some people, wig to them is like a battery. It gives them energy. I need to get one too. You know, they have one, they put it with glue over the head. Yeah, that's disgusting, but I don't know. Let me just go transplant. I'm going to go transplant in Turkey. I said that one time joking. My cousin went and did it. Okay. All right. All right, I got to keep my mouth to myself. It's $2,000. Everybody looks better when they get a transplant. I just don't have time to go. I'm always studying, preparing, but I will go one day. Okay. All righty. We talked about that's really important. Yeah. No. Okay. We're getting to that. That's a very important question. All enzymes, guys. That's a very excellent question asked by your classmate. Are all proteins enzymes? No. Many significant proteins are enzymes. All enzymes are proteins. Are all human beings police officers? No. But all police officers are human beings. But there is other profession, like some of them are doctors, some are teachers, some are, you know, all that stuff. So we will go over that actually today. So this graph is money. This graph is really important for you to understand. On the left is the Gibbs free energy. And on the right is the time of the reaction. So without the enzyme, this whole green part, look at the green part. Where does it come from? It's right from the level of the reactant up until who? The top of the hill is who? No. The energy is the activation energy. I'm saying what is that? Excellent. The activation energy is the energy from the reactant level up until the transition state complex. So all this green part. But if you look from the level of the reactant all the way down to the product, the pink one. So we have green energy and pink energy. This diagram here has a catalyst. We call it a biocatalyst because it's accelerating a biological reaction. Are all reactions in life biological? No. Some reactions are chemical. They're chemical reactions. So the ones who accelerate biological reactions, we call those biocatalysts. If they accelerate chemical reactions, we call them chemocatalysts. So if you look now, you will see that this is the activation energy and this is the net free energy. If you compare both, here we're not using a catalyst, here we are using a catalyst. What happens to the pink energy? Has it been affected? Is not affected. The energy from reactant to product is not affected. Only the energy from reactant up until the hill, the activation energy up until the transition state complex, that's the one that drops.
Is that still the free energy? Is that considered free energy?
So all energy here is free energy, meaning that that's the energy involved in the reaction. But we have different types of free energy. We have a free energy from reactants up until the transition state complex. That's the one that gets affected. And then you have the stationary, the stable energy between a reactant and a product. That's like the minimal amount from here to Staten Island to where you're going. That one, even if you take an airplane, even if you take a helicopter, is never going to change. What's going to change is the route you're going to take to make the reaction go faster. Are we clear? Now, how exactly are we able to achieve this lowering of the activation energy complex? Well, obviously, the reactant has to bind to the enzyme. You know, the enzyme is going to distort the bonds. The old bond between A and B is going to break it. The enzyme breaks it, literally breaks it, distorts it, and starts to form the bond between B and C. It's going to make a modification. So everything that is reactive, we call it a substrate. Why do we call it a substrate? We call it a substrate because the enzyme can target it. If you bind to the enzyme, you're a substrate. If you're a customer for the enzyme, but they don't call it a customer, they call it a substrate. So the enzyme releases reaction products. So the enzyme is unchanged and it can repeat the processes. So here the substrate sucrose is hydrolyzed. So you need to be familiar and comfortable with this word, hydrolyzed. You know what we're dealing with, hydrolyzed. So we're dealing with water. So that means somebody is donating an OH, somebody is donating an H, water's coming out, that is dehydration. A bond is formed. Hydrolysis, lysis, is when one donates an OH and the other donates a H, a hydrogen, and then water comes in, hydrolysis. Okay, everybody look, that's the enzyme. And what is that? The substrate. That is the substrate here. Once you bind it now to the active site, you need to know what the active site is. The active site is the location where the substrate is going to bind. So the substrate is going to bind to the active site. And then you're going to get the product where the product is going to leave the active site. You need to know what a substrate is, and you need to know what an active site is. The substrate is the reactant that we need to break the bond or maybe sometimes form the bond. That's the substrate. That's the customer for the enzyme. And then where does the substrate bind? It binds to the complex, the enzyme substrate complex. Are we clear? We talked about temperature and pH affecting the enzyme shape and action. So for example, you were asking earlier about your classmate had a good question about what is the temperature or what is the concentration, the specific concentration, the threshold that at that point we're going to get, a plateau or we're going to get a ceiling effect. Well, it depends. Salivary amylase is an enzyme in your saliva, in your oral cavity. You studied that in AMP1, remember? Pepsin is in your stomach. Both are enzymes, but do they work at the same pH? They both have a different optimal pH. So salivary amylase works best at 7, pH 7. If you change the pH, it's going to denature. It can never hydrolyze any any carbohydrates that you eat. You're eating pancakes, not broken down. But pepsin works at 7 pH. I'm sorry, not 7. Works at pH 2. It's very acidic. If you switch them up, you put amylase at pH 7. If you put amylase at pH 2, or you put pepsin at pH 7, then you rest assured that it's not going to work, because it's no longer the optimal pH. There's also an optimal temperature. Most enzymes, they have an optimal temperature of 37 degrees Celsius. Some of them have extreme temperatures, like bacteria. Some bacteria live in our body. Do you believe that they discovered some bacteria that live in the intestine, not of human beings, but in the intestine of termites? You know termites? What do termites eat? Wood. Some bacteria live there in the intestine of a termite, eating wood with the termite. So there's always some crazy type of temperature. Everybody has a particular set temperature, but some have extreme, like that bacteria who lives inside the termite. Yeah, so it depends. Some of them, the worms come in eggs. The person that eat meat, especially red meat. This meat, we're not the only one that love meat, the worms, the parasites love meat. No matter how hard you cook the meat, you're never going to kill all the worms and the parasites. You're going to kill a lot of them, and the immune system is going to fight the rest, but you're not going to kill all of them, for especially red meat. Chicken is a little better. Fish, what about pork? Fish is a little better. I mean, when it comes to pork, oh boy, that one is the king of worms. You cook it as hard as you can, it's not going to work. But anyway, my point is, the person who eats a lot of, the point that you can do anything is not going to die. Listen to me. The person who eats meat, they are bringing a lot of parasite in their body because they live there. But if you eat vegetables, it's a little better. Actually, some of them live in vegetables also, but not as much as meat. So now you eat meat, you're bringing in a lot of parasites to your body. So more parasites coming in, and the old parasites are sharing the meat with you. So you're not eating only. So they're in there. So they're eating with you. But when you're hungry, they die. When you're hungry, they cannot, you can handle starvation, but they cannot. So they're going to die. So the people who fast a lot, yeah, so when people fast a lot, they're like calm. This, the nervous colon, people who eat a lot of meat, they're always tense because your digestive system is connected, the interior nervous system, because when there's a lot of meat, digesting the meat requires a lot of nervous activity. So these people are always tense. But people who are vegetarian, for example, moderately, not like completely vegetarian, moderately vegetarian, or you fast a lot. So now your digestive system is not really tense. All of the parasites didn't get food, so they start dying. So now you're more healthy, you're more happy, you're more calm. Anyways, okay. Cofactors. Yeah, it's true. Yeah, so you if you don't eat a lot of meat and you don't eat a lot in general, you're gonna be so peaceful. All of the worms and the parasites are gonna die. All the devils, they're gonna die. You're not gonna have any devils. You're not gonna have any worms. Yeah, you're not gonna have any parasites or worms or devils in your body because you're not giving them any food. They die quickly. You can handle hunger, they cannot. Like Listerine, you can handle it, they cannot. Okay, all righty. That's a perfect example of nicotinamide dinucleotide working as a shuffler. So they go to the glycolysis. These are two separate reactions. Glucose catabolism to pyruvate and pyruvate carbon fixation. These are two separate reactions. Look in the middle. Nicotinamide dinucleotide NAD plus is obviously plus, just a cation. That means it lost an electron. That means it's been oxidized. That means it's a reducing agent. So it's going to accept an electron from one reaction and it's going to give it to the next reaction. If you do that kind of stuff, they call, they say the real estate broker, working like a broker, stealing money from you and stealing money from the person who's selling the land, and they're just shuffling in the middle. This one is like that. So it's performing this, and we call that a cofactor. Don't students mix up prosthetic group with a cofactor. No, a prosthetic group is a moiety that is attached to the enzyme, like iron, heme attached to hemoglobin. That's not a coenzyme. That is a prosthetic group. The coenzyme is working. The heme group is not working. It's a part. It's like a wig. It's like an extra leg. But the coenzyme is is working with the enzyme. Okay. Listen, metabolic pathways. Let's take a 15-minute break and come back. 15-minute break. 1-5. Let's come back at 6:25. 6:25. We continue. We have a lot today. Don't touch, please. The thing going on. I don't know how to stop it and then combine them both. I'm tired. So let's just let them. I'll see you guys. Pause it. You can pause it. You can pause it.
But if you're going too long, you turn off.
Yeah. Pause it. Yeah, pause
If you're
it.
going too long,
Are you a doctor?
turn
You
off.
should know that.
I'm telling
No,
you,
no.
right,
Pause it. Pause
if you're going too long without using it, you'll turn off.
it. Turn off.
Yeah, just make sure what's happening.
Okay. Yeah, I'll be back quick. I'll be back quick. Okay. All right.
All the thing is on there. It's still on there.
Okay, guys, remember exam, I want all 50s. It's out there for you. Before physics come. All righty, so let's continue. Now we saw that our enzymes are implicated in all of our metabolic reactions. Some reactions are catabolic, others are metabolic. But in the end of the day, we can never achieve a full completion without an enzyme. So A is a reactant, and B and C are intermediates, and D is the end product. So here you see that there are three different enzymes, alpha, beta, and gamma. Alpha, beta, and gamma are working together to take A. It's like a metamorphosis. The alpha is going to convert A to an intermediate B. B is going to be an intermediate C, thanks to the actions of enzyme beta, and C is going to be converted into the last finalized product, D, thanks to the activity of enzyme gamma. The metabolic pathways are turned on and off by the alternating enzyme activity based on your demand. Are you in a state of metabolic energy expenditure? Are you spending the energy? Then you're going to turn on the enzymes that are going to be favoring the catabolism so that the energy can come out and supply you with energy in order to be able to function. Are you in a state of laziness where you need to be storing the energy and not burning it? Then the other set of enzymes, enzymes are not all the same. Some do catabolism, some do metabolism. Some don't even do catabolism or metabolism. Some of them even are only responsible for structural change, structural integrity, like collagen and keratin and so on. Where is our good friend who asked earlier, are all proteins enzymes? Okay. Oh, you see, when we want to answer her question, she's gone. So all of these are different types of proteins. For example, look at A. A is a receptor protein. So this protein here is not an enzyme. This protein here is not a structural protein. That's a receptor. So it's responsible for sensing, detecting, and binding to a ligand, binding to a stimulus. That's its job. Some other protein is an enzyme. It catalyzes the breakdown of a particular ligand. Some other protein is not an enzyme, is not a receptor. What's going on in the back? So we have a third type of protein in purple, is not even a receptor, is not even an enzyme, is a channel, is like a tunnel. It allows for the entry, influx, exit, efflux, entry, influx, exit, efflux. That's another type of protein. That's a gated channel now. That one is only going to open if like the membrane voltage causes it to open. Some of them are ligand gated. So you have to bind some neurotransmitter like acetylcholine that causes it to open. And this orange one here is not an enzyme. It's not a channel. It is not a receptor. That's a cell identity marker. Who can tell us what is that? What's a cell identity marker? Can you say it again? I cannot hear. I couldn't hear a word you said. I just saw your mouth moving.
What type of the one that attacks what type it is that is carrying?
So what is it? This whole cell identity marker. What could that mean? What does that even mean? A marker, like a highlighter? What is that?
No.
So what does that mean? Exactly. So for graft rejection. So for example, can I donate a kidney to my identical twin? Absolutely, my identical, because we have the same identity marker. So when your immune system is doing immunosurveillance,
it's your identical
twin.
Because they
Yeah.
have the same identity marker.
Exactly.
But can the family member also do it? Because I get they mad.
Yes, it happens, but
I gave a kidney to my uncle.
that can happen, but it's not gonna, at some point, eventually, the immune system is gonna catch up. So for example, if I gave it to your uncle, the percentage, the speed with which the immune system is gonna do the way the immune system is gonna do its immunosurveillance. You know, your immune system is going around like scanning, exactly, scanning. So it's scanning those identity markers. That sounds like one of us. That sounds like one of us. That sounds like one of us. If it's a different identity markers, like the kidney I gave to my uncle, then that's gonna be detected as a foreigner, enemy. It's gonna get destroyed because it has no way of knowing that.
That's why they look for the match in the family, like it's my aunt and my mom did not match it. So I was the only one to match it.
Yeah, so that means your cell identity markers were closer more similar. What is going on in the back? Stop talking. Please listen. So the cell identity markers is what your immune system is responsible for detecting. Oh, this is one of us. This is one of, imagine there's a mom, she's blind, but she touched her baby, the nose, the eyes. Oh, that's my baby. That's exactly what your immune system does. It's going around scanning for identity markers. Now, some people share some identity markers. Only the identical twins, not even the fraternal twins, have the same identity markers. So if you have the same, sometimes the immune system has a disease, it no longer remembers the identity markers. So it passes by your ligaments and your joints, and it identifies them as, like in autoimmune arthritis, rheumatoid arthritis. What's going to happen is it's going to assume that the cartilage is like an enemy because it has only self, non-self. It has no way of knowing that, yes, this kidney is not one of us, but it's there to help us. Has no way of knowing that. It's either you have my self identity markers, you're one of us. It's like Trump. You're either for the US, you're good, you're not, you're bad. So the same, that work the same way. It thinks all foreigners are bad. Your immune system doesn't know that this kidney is a foreigner, but it's there to help us. Like some of these illegal immigrants, they're trying to help their families, but he has no way of knowing that. He's covered in Cheetos dust. Okay. Okay, what is cell adhesion molecule? Cell adhesion molecule is a staple. It's going to anchor, hook the cells together. That's how we have tissue. Your tissues are not still, your cells in your tissues are not sitting like the way you are sitting now. They are hooked to each other to give a strong tensile integrity for your tissue and it doesn't fall. You need to know the second messenger system, part of your proteomes. We talked about that. We said that what if you call the Uber and the Uber cannot walk into the school. So they talk to the public safety, then the public safety walks into you. That's the second messenger. But if the Uber driver happens to be a student of Helene Fulp, who has a job at night working Uber, that student, they take the ID, they come in. So if you look here, the plasma membrane is full of cholesterol. The plasma membrane is full of cholesterol. So only cholesterol-based hormones can pass, like testosterone, estrogen, progesterone, aldosterone. They're welcome to pass through. But if you look at this red triangle, a perfect example of that would be epinephrine, you know, adrenaline, fight or flight. That one could not cross because it's not cholesterol based. That means it is polar because cholesterol is non-polar. So it cannot cross the polar plasma membrane. So it's going to bind to the receptor. And is this a second messenger? No, that's a G protein who is going to go to adenylate cyclase. That's the enzyme. The enzyme is going to convert GTP into cyclic AMP. That's the second messenger. Students think that the G is the second messenger, adenylate cyclase is the second messenger. No, they're not. Only adenylate cyclase is going to convert ATP into cyclic AMP. That's adenine triphosphate. That's adenine monophosphate that is cyclicized. That's the second messenger who's going to go activate a kinase. We're going to learn today that kinases would add a phosphate group. And the kinases are going to get activated, and then they will go and they convert some inactive enzyme to an active form. And that's going to lead to the target effects. The target effects, various metabolic effects. Some proteins, that's what we call the glycocalyx. The cell identity marker is a glycocalyx. It's like a protein attached to some lipids, protein attached to some sugars, glycolipids, glycoproteins. Some proteins are structural, like the cilia. You know, cilia to remove all the mucus that is building up in the trachea. the cilia. Also, these are the cilia. These are like the bathroom mop. They keep mopping. They have two stroke: power stroke, recovery stroke. Power stroke, recovery stroke. Power stroke to shove and move the mucus. But in a disease known as cystic fibrosis, you need to know that there is a mucus plug. There's so much mucus that is overwhelming to these particular to the cilia. Alrighty. Pseudopods. Pseudopods is cell crawling. You know, cell need to walk. Some cells need to walk. Not all cells need to walk. Who can give me an example of a cell in your body that is all about walking? A walking cell. Don't say a red blood cell, because a red blood cell doesn't walk. It swims. I need a cell that walk in the tissue. Chemotaxis. Taxiing by detecting chemicals. Oh no, our class of 60-something people has no idea of an example of a cell in your body that walks. Think of a walking cell. You're not a cell, you're an organism. Huh? Yeah. What about white blood cell in the tissue, looking for bacteria? Yeah. So now that white blood cell, yeah, is going to have a lot of pseudopods. Pseudopods to help it crawl in the tissue, walking around. Is there any bacteria here? Any parasite there? Any particular fungi? We need to fight it, combat it, phagocytosis. So in order to walk around, it's going to need pseudopods. Pseudopods are proteins. So all that is showing you the involvement of proteins in the cilia. There's also flagella. So if you see here, these endothelium, you see that there are clefts, gaps, interendothelial, meaning between the endothelial. There's gaps that allow for the proteins to pass through. We talked earlier about the ceiling effect, the plateau. It's when there is a transport maximum. All the shuttle buses are full. So now it's not about concentration no more. In the past, it used to be, you see, the more concentration, the rate of the transport is faster. So the more, the better. The more, until you got to a point where it reached a plateau. That's the ceiling effect. Okay, that shows you the actions of proteins on the plasma membrane to aid in diffusion. Like, for example, there's too much glucose outside the cell. It's going to diffuse down the cell, but glucose is polar, and the plasma membrane is non-polar. So the glucose cannot cross. So it needs a facilitator. It needs a protein to facilitate its passage from outside to the inside. And there's the involvement of proteins in secondary active transport, where you see a lot of proteins hitchhike. So here, a lot of sodium is coming in. Glucose says, can I come with you? So you see both of them are influxing. And here you see that the sodium is coming out. Then sodium is removed outside the cell to restore this gradient. The sodium potassium ATPase, the greatest, most important enzyme in your body, because this enzyme is the one that is able to restore the gradient to ensure that there is no balance between extracellular electrolyte concentrations and the intracellular electrolyte concentrations. You see that they're all different. And that's why if you get bitten by a snake, God forbid, the venom of the snake, this digoxin is going to shut down the sodium potassium ATPase. So what happens as a result? You're never going to maintain a concentration gradient. Sodium and potassium inside the cell intracellularly and outside the cell extracellularly is going to be equal. So now you're never going to be able to make an action potential. No being able to make an action potential, meaning your heart is not going to beat. You're not going to, you're going to have a shock, drop in blood pressure, and death. That's how the snake kills you. Okay, the movement of proteins through the cell is known as transcytosis. Some proteins, those are the forgotten proteins. We all think that proteins are like are some enzymes. Some proteins are not enzymes. They're not protein channels. They're not cell identity markers. They are not cell adhesion, hooking up the cells together. They are scaffolders, like the cytoskeleton. That's why they say skeleton, because the cell is held with a scaffold. These are like the actin, myosin, the microtubules. Remember mitosis? How in mitosis you have some chromosomes that are lined in the middle, then they are pulled apart. Who does all of that? Structural proteins, microtubules, push them all the way. Microtubules push the chromosomes to the middle and they pull them apart. So if you look at the involvement of cytoskeletal proteins, this is very important. I hope I got it. Okay, cytoskeletal proteins. Those are proteins that are all about holding up the building, holding up the cell from collapsing on itself. So an example of that, the best involvement is in cell division in mitosis. In mitosis, you have the centrioles. Those are the centrioles. Centrioles at the two opposite poles. The centrioles. The centrioles are going to start pushing. They're going to start pushing the chromosomes away. They're going to be pushing the chromosomes in this direction, pushing the chromosomes away until they get to a point where chromosomes now have been pushed by these centrioles. Chromosomes have been pushed by these centrioles all the way to the middle. Chromosomes have been pushed by the centrioles all the way to the middle, and now all the chromosomes are now lined up in the middle like that. So who pushed all these chromosomes to line up in the middle? It was these microtubules. Then, when it comes to the cell division, those same centrioles are now going to be plucking off and pulling away all of the chromatids. You see, one time they pushed them toward the middle. They pushed all of the chromosomes toward the middle until they lined up in the middle at the metaphase plate. But once we reach anaphase, now they're going to pull in the opposite direction. They're going to pull in the opposite direction until we pluck off all of the chromosomes, and now you're going to have something like this. You're going to have something like that where until then you have two separate cells. So that shows you the involvement of these microtubules, the involvement of these too small tubules, these tubes that pull apart all of the chromosomes in order to help in cell division. So if there's no cytoskeletal proteins, we're not going to be able to do cell division. We're not going to be able to grow. We're not going to be able to develop. It's never going to be growth and development of the organism. So that's your cytoskeleton. So not only does it help, so if you look where my mouse is, you see here, those are the centrioles. You see the centrioles? The centrioles is where all the microtubules originate. They come out of there. And you see they are pushing away like a scaffold, like construction. They're holding all the integrity of the cellular structure. Are we clear? So what is that? That's a microtubule. Where does the microtubule come out of? Centrioles. Very good. And you see, those are the microtubules. They are made up of a protein known as tubulin. Tubulin. So many tubulin proteins give you a microtubule that is made up of multiple protofilaments. And then there are additional structural proteins known as dynein arms. All of this is a protein structure that is responsible for structural support, scaffolding, and structural integrity. We talked last week about many proteins that, 2 weeks ago? The shredder, same thing. The shredder, don't be too literal. See what I mean with my word. Last week, last meeting, 2 weeks ago. So that's a shredder. The proteome is the shredder of proteins. Why do we need to shred proteins? Because the proteins are misfolded. Remember mad cow disease? When the cow starts to develop misfolded proteins in its brain, people who snort cocaine and heroin, they have a large rate of turnover. There is a lot of misfolded proteins in the brain, and that overwhelms the proteasome. You need to remember the proteasome is a shredder of all the misfolded protein. How does the proteasome know that this protein is supposed to be shredded? And why doesn't it go to any random protein and shred it? Why does it know exactly it needs to shred a particular protein? Look in there. There is a reason, there is a particular marker that the proteasome, when the proteasome sees that marker, when the proteasome identifies the purple marker, do you see a purple marker? When the proteasome sees that purple marker, that's the target misfolded protein. That's the messed up protein. That's the one that's going to give mad cow disease or Creutzfeldt disease. It gets tagged with a purple tag known as a ubiquitin tag. If you add ubiquitin to any protein, the protein is going to shred it. Are we clear? Okay. You see, these are the centrioles, these are the microtubules. And you know something very special about proteins and where they're made. Proteins are made in the ribosomes, but there are two types of ribosomes. There are ribosomes embedded on the endoplasmic reticulum, and there are ribosomes that are free floating in the cytosol. What's the difference? What's the difference between ribosomes free floating in the cytosol and ribosomes that are embedded on the endoplasmic reticulum? If you look here, that's the rough endoplasmic reticulum with all these dots. All these dots. Those are all ribosomes. Those are all ribosomes. And that's the smooth endoplasmic reticulum. It has no ribosomes. Ribosomes don't break anything down. Ribosomes make proteins. So how are the proteins that are embedded on the endoplasmic reticulum different from proteins that are free floating on the cytoplasm? Well, in the end of the day, they're all proteins. We have two types of ribosomes. We have two types of ribosomes. What are the two types of ribosomes that we are concerned with? So we have ribosomes. Ribosomes. There's two types, type 1 and type 2. There are ribosomes that are on the rough endoplasmic reticulum, RER, rough endoplasmic reticulum. And there are ribosomes, free floating on the cytosol. Well, two types of ribosomes, but in the end of the day, they are ribosomes. And no matter what kind of ribosome are you, you're going to be making proteins. So the only difference is going to be on what type of protein that are being made. So the rough endoplasmic reticulum is going to make some proteins that are to be used outside the cell. You know, you have a restaurant and the same kitchen. In the same kitchen, they make food for the customers and they make food for the workers and the waiters and the chefs themselves. I don't know which one is better, but there's two different kinds of food. One is going to the waiters and one is going to the people, the customers. Same thing. In your cell, there are two types of ribosomes. Ribosomes that make proteins for its own use, to be used locally inside the cell. And some ribosomes are going to be making proteins to be used outside the cell. Who's going to be making proteins to be used outside the cell? The rough endoplasmic reticulum bound protein. So after the ribosome makes the proteins, the rough endoplasmic reticulum adds a tag, like a barcode, adds a tag known as the signal peptide sequence. Signal peptide sequence. Signal peptide sequence. But the ribosomes that are free floating on the cytoplasm, they don't have a barcode. So all the proteins they're going to be making is going to be used inside the cell. But the proteins that are made by ribosomes that are embedded on the rough endoplasmic reticulum, they will have this additional signal peptide sequence. This signal peptide sequence is going to go to the Golgi body. Is going to go to the Golgi body. And the Golgi body is whenever it sees that signal peptide sequence, is going to know that's the international shipment. You know how in Amazon, in the warehouse, they're going to look at a barcode and they're going to be able to say, oh no, this is not from state to state or from border to border, is going out the country. Same thing. Any protein that has a signal peptide sequence, the Golgi body is going to receive it and package it. The Golgi body is the Amazon warehouse. It's going to package it and export it outside the cell. Export through exocytosis, outside the cell. Outside the cell. Clear? Now we know how are different proteins made. If you're to be used inside the cell, where do we make you? In the free-floating ribosomes. If you are to be used outside the cell, we make you in the rough endoplasmic reticulum. Okay, so the ribosomes, we make them, and this is the Golgi body. As you see here, the Golgi body has an entrance and an exit. The entrance is the cis side. The exit is the trans side. It looks like a pita bread. You know, multiple pita breads on top of each other? That's the Golgi body. This is the packaging warehouse. You see, I love this diagram here because that shows you what I was just talking about. What is that here embedded in these ribosomes? What is this structure here in blue? That's the rough endoplasmic reticulum. If you say ER, there's two types. There's the smooth ER and the rough ER. That's the rough ER. Now look at the rough ER. You have proteins formed by ribosomes on the rough endoplasmic reticulum. So now you know they have a signal peptide sequence. So now they're going to be leaving the rough endoplasmic reticulum and they're packaged into transport vesicles. Then they're going to go and they're going to fuse into clusters to form The cis is the entry site. You see now they have all entered into the Golgi body. The Golgi body is going to modify them and repackage them, and it's going to exit through the trans site. The entry site is the cis. The trans site is the exit site, and you see now they are exported through exocytosis. See, all the proteins are now leaving, exiting the cell. Is that clear? The glycocalyx, this is your cell identity marker. It's a combination of proteins and sugars, glycolipids, and proteins and fats. Glycolipids, glycoproteins is a combination of surface. Always cell identity markers got to be at the surface. No cell identity marker is hidden somewhere inside the cell. So you have glycoprotein, sugary proteins, or glycolipids, sugary fats. Those are the combinations that your immune system is going to screen and scan to tell self from foreign or non-self. And that's another example of proteins being the microvilli used in the absorption in your intestine to increase the surface area. And you have some structural proteins that have a job of absorbing the food that you broke down in the intestine, whether it's lipids, proteins, carbohydrates, and through the microvilli absorbing it into the blood. Cilia, we talked about cilia for movement. So you see that the power stroke and recovery stroke to push the mucus. Cystic fibrosis is when there's a mucus plug caused by hereditary mutation in which chloride pumps are not there. So the chloride pumps fail to create an adequate saline layer. So there's no chloride, so water is going to build up. So the thick mucus plug is going to be plugging the pancreatic ducts and also the respiratory duct, overwhelming the cilia. And there's inadequate digestion of the nutrients, and there's inadequate absorption of oxygen. And the mean life expectancy of cystic fibrosis patients is 44 years old. Proteins are also implicated in flagella. So you know flagella is found in only two places, bacteria or the sperm. The sperm uses the whiplash movement in order to move and swim in a mucus fluid in the vaginal tract, ascending up into the uterus. So it needs the flagellum that is powered with a lot of mitochondria in the neck region so that the tail can move fast. We talked about microfilaments and microtubules in the cytoskeleton. Now, the last thing we need to know about the proteins is that we need to, how do we learn everything about the proteins? How do we know that there's some structural proteins, some functional proteins? We need some laboratory techniques that we perform in the lab that help us understand the structure and the function of these proteins. We're going to go over them now. So we open the cells, we fragment the cells, we break apart the plasma membrane. We homogenize the plasma membrane so that we can leak out the intercellular content of the cell. Then we centrifuge. Centrifugation is going to separate for us the content of the cell based on mass. When you centrifuge a test tube, you're going to see different fractions. The heaviest is going to sediment all the way in the bottom, and the lightest is going to be all the way at the top. So another mechanism is dialysis. We're going to use pores. We're going to use a barrier with different holes. And now we're going to separate proteins based on size. So how big are these holes? We make them with certain sizes, and any protein that leaks out, that means it was able to make it through the holes and the tunnels. And now we can tell the size. Hemoglobin is of this size because it was able to make it through the pores of a particular size. That's known as dialysis. When you separate salts from proteins and you separate particular structures based on molecular mass, not charge, not chemical structure, but only based on the size. Chromatography. In chromatography, we separate based on different variables. So those are different types of chromatography. You need to know, chromatography, we separate proteins based on different structure. You know, if you look at people, people can be divided based on age. You can have a group that is pediatric, a group that is adult, a group that is geriatric, really old. They could be separated based on the sex. There are gynecologists, obstetricians, and there are males. Sometimes we separate based on the different criteria. Proteins also can be separated based on different criteria. Remember, I was saying the R group has different criteria. If I want to separate the proteins based on the charge, I want to tell if this protein positively charged or negatively charged. I can use ion exchange. So I run my protein through a positively charged medium. If it makes it through, that means it is positively charged, because positive repels positive. But if it doesn't pass through, that means it is a negative charge. So I come up with a technique. I have a mysterious protein. I don't know if this protein is positively charged or negatively charged, but I know my medium. My medium is positively charged. So I run my mysterious protein through my positively charged medium. If it makes it out, that means it was repelled, because same charges repel. But if it gets stuck in the medium, doesn't come out, now that's how we found out that some amino acids are negatively charged, like glutamate, aspartic acid. Why? Because it never came out the medium that was positive, because it bound to the positive. We have size exclusion, gel filtration, where we run our different protein of mysterious size through beads of known size. If it makes it through the medium, that means it's smaller, because it was able to zigzag and pass through. If it gets blocked at the top, that means too big to pass through the holes and the pores. That's another property, looking at the size, separating. So here we didn't separate based on charge. We separated based on size. Affinity, that's what I was saying earlier. There's a binding using the charge. Affinity, meaning you have a negatively charged medium. If your protein gets stuck, that means it's positively charged, because only opposite charges attract. Separation based on polarity. An example of that would be the reverse phase chromatography. In the reverse phase chromatography, we don't know the protein has which polar end. Which end of the protein is negatively charged and which end of the polar of the protein is positively charged. So the protein has two terminals. It has a carboxylic terminal and it has an amino terminal. So we run it through the chromatography paper to find out the polar side. Is it this particular mysterious side? Is it positively charged or is it negatively charged? Now, that shows you the ion exchange chromatography. We went over that, where there's cation exchange. So here you see these beads, those little crystals are charged. There's anion exchange chromatography, where the beads are negatively charged, and cation exchange, where the beads are positively charged. So you look here, you put a mixture of unknown molecules. You see how tiny these beads are. So you see that the small molecules are going to squeeze and move through the channels in the beads, and they're going to elute later. But the large molecules are going to zigzag through. You see those beads have little holes on them. So when you pour the whole solution at the top, they are too small, they get trapped in the little holes. But the really purple, if you look at purple and red, can everybody see purple and red? Purple and red was a mixture of proteins. We don't know who's big. We don't know who's small. Our medium are these light blue beads, but those beads themselves inside them have really, really tiny holes. So when you pour red and purple on the top, you will see that the purple is so big to go through the holes inside the bead. So it will bump on the outside and get out and move out. It's going to elute. It's going to come out the end. While the really small ones is going to take them forever to come out. Why? Because they are now moving through these holes, while the big particles are not able to fit in through, so they bump and move faster. Is that clear? Is that clear? Do it one more time. I have purple and red. That's a mixture of protein of an unknown size. So I pour everything at the top here. You see these blue beads. Those beads are not balls. They're not just circular round ball. There are so little tiny passageways inside them. You see the passageways? They're so tiny. They're tiny that when you pour everything, the red particles are going to go through the hole. So they're going through like a maze. You know the maze? So they're going through, they're too busy going through the maze. The purple particles are so big, they cannot fit through the beads. So they just bump and they just move and they zigzag and they come out. While the small tiny particles are not going to come out because they're too busy moving on the inside, in the inside of the maze, in the inside of the tunnels. But the tunnels were so small that the purple ones could not bind to it. So they were just zigzagging by. Is that clear now? Got it? Okay. So large molecules are going to zigzag and pass through, and tiny molecules are going to be attracted to the inside of the bead, and they're going to be passing through the bead. But the purple one is not going to pass through the bead. It's too big. It's going to be passing between the beads. Is that clear? That's affinity chromatography. So here I have a particular type of protein, and I tag the protein with a nickel column, and then I have a particular filter for me to, is like a magnet where I need a particular histidine tagged protein. I know histidine is plus, so I add the imidazole. The imidazole is going to be attracted to the histidine. So you will see that my filter is going to trap with affinity, like a magnet, is going to be attached to my protein of interest. In this case, is histidine tagged. And everything that doesn't have histidine on it, that is not part of my filter. So it's going to be other unwanted protein is going to leak out, while my protein of interest is going to be, this one is the final product that I got with my filter. So I put a filter for an affinity of my target protein that I want from the mixture. I have a whole I have a salad. I have a different salad of all different proteins. I don't want all of them. I want a particular type of protein. So I tag it with histidine, and I put on the filter an affinity for histidine. And then I pour everything. Only my protein of interest that is tagged with histidine is going to bind to my filter paper. This is reverse phase chromatography. And here we use the electricity in order to separate and find the two poles, two opposite poles of my proteins of interest. Agarose gel. Here we separate based on the charge using an electric field. So you see these holes here, they are holes of agarose gel. I take my pipette and I put my different fragments. I have chopped up the protein. It's used for both protein and DNA. And I put them in different holes, and then I run the electricity. The smallest fragment is going to move really fast. and it's going to be at the forefront, while the slow ones are going to be because they're so big and heavy, they're going to lag behind. So now I'm going to be able to tell my protein is made up of which particles. Okay, loaded in wells, we went over that. The largest molecules move the slowest. There you go. You see here, the heavy ones are in the back and the lightest ones move all the way to the front. Those are my loading gel, and the smallest ones move to the front really fast, and the heavy ones get stuck to the back. So the largest ones are heavy. They stay in the back, and the lightest ones move because they're so small fragments, they move to the front faster. Okay. Western blotting. It is useful for identifying proteins that are found in a gel. So this is very similar to agarose gel, and I use the same concept, but Western blotting is reserved for separating proteins, only proteins, while SDS page, this one, is used for both proteins and DNA. But it's the same concept. And in the end, when I separate my protein, I rely on antibodies that are primed to tag my protein of interest. And then I use like a magnet. This time I'm not using a charge. I'm not using a filter paper. I'm using actual antibodies that I have marked them to go and tag for me my protein of interest. Okay, we talked about enzymes, substrates is the target for, and the active site. Rather remembers enzymes catalyze the reactions. Substrates are the customers for the enzyme. They bind to the active site. And you see, this is the active site. You see, this is the active site. And what is here in black? That is the actual substrate. And this is the active site. So in the enzymatic reaction, I have reactant A and B, and that's my enzyme. Substrate A and substrate B, both of them. So that's an example of a multiple reactants. They're going to bind to the active site. And you see the enzyme is going to distort the bonds. And you're going to see that there's a transfer of this purple group. So you have an enzyme ES, enzyme substrate complex. So the enzyme is going to change the result. And you're going to see that there's a distortion. And you see that this particular moiety is going to be transferred. You see it moved from A into B. So A has now become C. And once you added this moiety from A into B, B has now become a product known as D, because it's no longer the same. So now that is EP. Before, you had the enzyme substrate complex, ES, and now you have the enzyme product complex, EP. ES, meaning we had the original structure, substrate A and substrate B. But that's the enzyme substrate. We're just about to begin the reaction. You have your enzyme and your substrate binding together, ES. Once the reaction went on, now you have the EP, which is the enzyme product complex. I have distorted, I have changed the structure of my original substrate, and now A is no longer A because you removed the purple part from it. So now we call it C. It has been changed completely. And B has now received this purple moiety here, meaning that it is no longer B, it now has become a product. So it's now becoming D. So we went from A and B into C and D. So that is our EP, that's our enzyme product complex. Questions? All right. So substrates affect the enzymes on binding. That's the enzyme plus the product. And you see, we go through a whole cycle. First of all, the enzyme, then bind the substrate. There are two separate moieties. The enzyme is separate from the substrate. Now both bind, the enzyme and the substrate bind together. And then you have the actual reaction of transferring and breaking and distorting some structures to go from reactant to product. And finally, you have the enzyme product complex, and you don't want the product to be stuck to the enzyme forever. So now we're going to separate the enzyme from the product, and now we release our product of interest. So the enzymes are known to have a high binding specificity. The enzyme is flexible because it's able to undergo some conformational changes to accommodate these different substrates. And the isoelectric point of this aqueous solution in which the enzyme is bathed in. So that's why we have an optimal pH and we have an optimal temperature in order to facilitate for this enzyme substrate interaction. So if you see here, again, the enzyme has an active site and purple is the substrate. So the substrate is going to bind the active site. So there's two models. The Fisher model says there's a fixed enzymatic active site. So we bind like a jigsaw puzzle. But the Koshland model says no, it is not a fixed binding between the substrate and the enzyme. The enzyme undergoes some changes. There's an accommodation. So you see the active site adapt to the substrate. So these are two different models that shows you how the enzyme is able to bind the substrate. Fisher tells you it's like a jigsaw puzzle. They fit in like two pieces of a puzzle. But the Koshland says no. The enzyme is able to undergo an adaptation, an accommodation to accept and receive the proper structural conformation to bind to this substrate of interest. So you see here, this is what is blue. What is blue here? That's the substrate. And where is the blue going to bind to? That's going to bind to the active site. We talked about that earlier. This is our transition state complex. We're going to drop it. We're going to lower the activation energy barrier. And we're going to see that the total free energy of the reaction is never changed. It is only the activation energy that is changed. So enzymes lower the activation energy. Enzymes catalyze reversible reactions. They do not change the overall energy. We talked about all of this. Single substrate giving you a single product. We talked about that. Single substrate giving you multiple products. Multiple substrates giving you a single product. And multiple substrates give you multiple products. So that's the lineage. First, this is the actual chronology of events. You have the enzyme going to bind a substrate. Then they actually bind. First, they were separate. Then they actually bind the enzyme and the substrate. Then the reaction takes place in the active pocket, in the active site. And finally, you have the enzyme converting the substrate into a product. Then they separate. The enzyme is separate from a product. This is the mechanism. So this shows you that the enzyme is going around. So that's what we were saying earlier. The more substrate you add, can everybody see where my mouse is? That shows you what we were saying earlier in the kinetics. If you have a little bit of substrates, the reaction is not going to go fast. The more substrates you add, you see the high substrate, if you add more substrate, the faster the reaction is going to go. Until you reach a plateau. You see that? Until you reach a plateau. When the Michaelis-Menten constant, Michaelis-Menten tells you that there is a constant known as the Km. The Km represents the substrate concentration at which half of the reaction speed has been achieved. So that is a marker that your friend was talking about earlier. Do we have known thresholds? So each reaction is going to have a different threshold, but there is the concept of the Km. You need to know that. The Michaelis-Menten constant Km. What does Km represent? Km represents the substrate concentration. So the Km represents the substrate concentration that we're adding. If you look at here, that's the key here. So in the beginning, you had a lot of shuttle buses, not enough passengers. Now, the more you add, the higher the substrate, the more action we get. But we all agreed that it's not the more you add, the better, because you're going to reach a point where you reach Vmax. Vmax is the ceiling effect, the plateau. Now, it doesn't matter how much more substrate you add, it's really not going to affect the activity. So now they came up with another constant known as the Km. The Km is the concentration of any particular reaction substrate. But if they tell you the Km for this reaction is 250 millimoles, you know that this is the reaction at which I got halfway before I reach my plateau, my Vmax. That's the maximum speed, maximum velocity, Vmax. So how do I know that I'm halfway to reaching Vmax? Is at the Km. So every reaction is going to have a Km that is different from the other. But you know, Km is the concentration of the substrate at which half of Vmax has been realized. Is that clear? Any questions? So Km is a substrate concentration that gives half of Vmax. And the enzymes that don't follow Michaelis, some enzymes don't follow Michaelis-Menten kinetics. They include the ones that bind to the concentration. Vmax occurs when the enzyme is saturated. Vmax is when all the enzymes, all the shuttle buses have been full. So Vmax occurs when the enzyme is saturated by the substrate, and Vmax varies with an amount of enzyme used. Km is a measure of the enzyme's affinity for its substrate. So the Km value is inversely related to the affinity. You need to know that. That's one of the questions that you will find in the exam or in the homework, that Km value is inversely related to the affinity. The higher the Km, the lower the affinity. So if I tell you the Km is high, that means the enzyme let go. The enzyme has let go of the substrate. If the Km is low, that means the enzyme is really bound to it means that the enzyme is really bound really tightly to the substrate. So the higher the Km, that means we're about to get to Vmax. So that means the affinity is low. That's it. We're gonna let go. We have too much of substrate now. There's no affinity between the enzyme and the substrate. Okay? There's not enough affinity between the enzyme and the substrate. You need to know this. That's one of the questions. The higher the Km, the lower the affinity. The lower the Km, the higher the affinity. Makes sense? Because the higher the Km, that means we're going to get to Vmax. So we're not going to have a high affinity for our substrate. The reaction is going to reach completion. But when the Km is low, we have a high affinity for our substrates. So if you look here, so the lower the Km, you see there's a little bit of substrate. Km is low. That means we're going to have a lot of saturation and binding affinity between the enzyme and the substrate. But when there is a lot of substrate, means the Km is so high, then the affinity is going to be low because all of the enzymes have already been saturated in the active site. Clear? Okay, the concept of enzyme inhibition. So the active pocket, the active site is where the substrate is going to bind. Let's say I want to shut down an enzyme. That means I need to block the active site. So those are known as competitive inhibitors. What is here in gold? What is gold? Gold is the actual substrate binding to the active site. A competitive inhibitor is gonna be like gold, but it's gonna be black or green or purple or some other color that is gonna fill the active site so the substrate doesn't bind. So I don't have a reaction. We use that in medication, like the ever heard of ACE inhibitors? What are ACE inhibitors? ACE inhibitors, they inhibit the enzyme angiotensin converting enzyme from working. So the medication is going to go to the active site of ACE, angiotensin converting enzyme, and bind it. So when angiotensin comes, there is no angiotensin converting enzyme. So angiotensinogen 1 is not going to be converting into angiotensin 2, and therefore our blood pressure is not going to be going higher. It's gonna drop low. So that's why it's a medication for anti-hypertension. But you need to know that competitive inhibition, you compete. That's why they call it competitive, because it competes on the active side, binds the active side. The non-competitive, you see the competitive inhibitor? The competitive inhibitor looks just like the normal substrate, and it binds on the active site. But if you look at the other type of inhibition, you have the non-competitive, where it doesn't actually bind or fight with the active site. It binds on an allosteric site and causes stoppage of the inhibition. Are we clear? It's called allosteric site, not the active site. It's an allosteric site. So another thing you need to know, if you want to overcome, how do you overcome competitive inhibition? The more gold you add, so because everybody's fighting over the active site, right? So if you want to overcome green, you add a lot more gold, and then the more gold is gonna overwhelm the more green. But can you use the same thing here with non-competitive? No, because it doesn't even fight on the same spot. You can have billion of gold. All we need is the non-competitive inhibitor binding to somewhere else that doesn't need to compete with it. So the most effective is the non-competitive, because you cannot overcome it with an increase in the substrate concentration. This slide shows you the different ways enzymes work. Some of them are oxidoreductases, so they're all about oxidation reduction. Are all enzymes working with oxidation reduction? No. Some enzymes are known as transferases. So they transfer one group, like hydroxyl, one of those functional groups, hydroxyl amino group, phosphate group, they transfer it. So that's not oxidation reduction. Hydrolases, they hydrolyze bonds. Lyases, they break bonds, but with a non-oxidative mechanism. Isomerases, they make an enantiomer that is different in the structure than the actual substrate. And lastly, we have ligases, like glue. They glue up, ligases. So they join two molecules by making covalent bonds. So that shows you E1, 2, 3, 4, 5, 6. It shows you that all enzymes don't work with the same mechanisms. Some enzymes work with blue, some enzymes work with green, others work with blue. So that shows you the examples here. So look at oxidoreductases. They began with a positive charge and they gained an electron, so it became NADH. So that's the oxidoreductases. It engages in oxidation reduction. Transferases, they have nothing to do with oxidoreduction. They transfer. You see, this molecule here had no phosphate. This enzyme hexokinase stole one phosphate from ATP. Now ATP was adenine triphosphate. It now became adenine diphosphate. And that phosphate was transferred from ATP to who? To glucose. Can you see it? It's right here. Was this has anything to do with oxidation reduction? No, that's a different mechanism. Now you see hydrolases. Those are the ones that break the bond. You see, proteases, they hydrolyze with water. They broke this peptide bond. Now you have two separate amino acids. And you have ligases, they ligate and glue up. You see, isocitrate, you see that you glue the glyoxylate to succinate, and then you end up with so liases, I'm sorry, I thought this is lyase. This is not a ligase. Lyase is when you break without hydrolysis. So you broke isocitrate here into glyoxylate and succinate, but you didn't break it with water. It's not hydrolysis. Isomerases, you see they make a different structure. So glucose 6-phosphate is now fructose 6-phosphate. They have the same number of atoms, but they are structured differently. Like 1, 2, 3, 4, 5, 6, 1, 4, 5, 3, 2, 6. So the same numbers, but they're ordered differently. This is known as rearrangement or isomerization. It's the same number. So fructose, glucose, galactose, they all have the same number of carbons, but they're all rearranged in a different structural isomerization. Ligases are glue. They glue up and they bind together different types of, they glue up and they bind together different types of molecules like we see here. Okay, any questions? That was we're done with proteins.